Thanks, Will. Thanks for the introduction. Great to meet you all. So yes, I'll talk today about one of those key sources of methane emissions in the United States and around the world, which is enteric fermentation. So I think for most folks, this first slide, right, is kind of very basic review. You're well understand the methane inventory in the U.S. and probably globally in terms of what those key sources are. In the US, according to the US EPA inventory, enteric fermentation is now the largest source of methane emissions. It bounces around year to year, right, in terms of the cattle inventories in particular. But it is the largest source and a big reason of why we are focused on it here at Colorado State University AgNext. So a little bit of quick review when we think about enteric methane emissions and just knowing there's a diversity of backgrounds on the call. To set it up, if we think about cattle, right, and this lucky cow that lives on Point Reyes National Seashore, so that is a real photo. We think about cattle, sheep, goats, deer, bison, all these ruminant animals. They have this unique structure in their stomach, right? This 4-compartment stomach, the largest compartment, which is the rumen, which is basically a microbial bioreactor, right? We can kind of think of there's trillions of microorganisms that live within the stomach of these animals, and they ferment feed. So, of course, as this cow is demonstrating, right, able to consume fibrous material, high cellulose material, and those animals are able to use the microbial fermentation that's taking place within their stomach to actually extract energy and nutrient value from what would otherwise be human inedible and inaccessible plant biomass in the human food system. So this little cartoon image that popped up, kind of the representation of what the ruminant stomach looks like. This largest component is called the rumen, the reticulum, kind of honeycomb structure, the omasum, and then they have a structure in their stomach that's called the abomasum, which is similar to our stomach in terms of an acidic environment. But the other compartments of the stomach are not acidic. They are where a lot of fermentation is actually taking place. So when those animals consume feed, it makes its way into the rumen, and then there is fermentation processes that are taking place, mostly bacteria, but there's basically all forms of microorganisms that are within the rumen that are taking through a variety of pathways that feed primarily to volatile fatty acids. Those volatile fatty acids, the 3 main ones that are produced within the rumen are acetate, propionate, butyrate. They get absorbed through the rumen wall. They can be used for the animal's own metabolism and energy purposes. The byproduct of all that fermentation, of course, there's a tremendous amount of CO2 gets produced, and then hydrogen or reducing equivalents that get produced as well in the rumen. Gases such as CO2 and methane, they get formed primarily from these substrates via methanogenesis, accumulate on the top of the rumen, and then roughly once per minute, there's a rumen contraction that will expel the methane from the animal's mouth on a regular basis, right? So that's the key thing, is basically nearly all that methane is coming from the front end of the animal. As I'll discuss, when we measure methane emissions, we typically are just measuring front end emissions, because that's where basically 98% of the methane is being released from the animal from its mouth. So there's some different pathways in terms of how methanogenesis takes place within the rumen. The key pathway, the one that we believe is the most important, is that hydroneotrophic pathway. But methanogens, different types of methanogens, can use different substrates, reducing equivalents, methyl compounds, acetate. And as we keep learning more, we're kind of understanding the relative importance of those different methanogenesis pathways in different cattle diets. So anyways, that's a high level. That is what enteric fermentation is. It's always kind of a little bit of a misnomer. It's actually the methanogenesis that's causing the methane, which is not a fermentation process, right, per se. More context here. If we think about within the United States, from 1990 to 2021, looking at the EPA inventory, the estimated enteric methane emissions are coming from different classes of animals. In the United States, enteric methane is dominated by cattle, right? Primarily beef cattle, as I'll share in the next few slides. It'll be clear why, and also dairy cattle. You also note this kind of pattern in the inventory, kind of ebbs and flows. A big part of this is a reflection of the number of cattle in the US, so it's not a constant number. There are fluctuations in the number of cattle, particularly beef cattle. In the long run, since the 1970s, the number of cattle in the US has actually dropped quite a bit. So we have about 40 million fewer cattle today in the United States as we did compared to the mid 1970s. What's interesting is, and this is what's accounted for in the EPA inventory, our animals, though, are more productive, meaning they produce more milk, they grow faster, they get larger. So we produce more human food per animal from our cattle herd. And a byproduct of that is those animals tend to eat more feed. So each animal is making more methane today as compared to 1990. So partially why we don't have this methane emissions reflect perfectly to the inventory of cattle is because each animal is producing more methane. Right? That kind of gives you an overview of recent trends in enteric methane emissions. The reason, again, we're so focused on it is even if we take a broader view, a life cycle assessment view, and we looked at the carbon footprint of milk, for example, on this slide. So this is a partial life cycle assessment looking at basically all the impacts, the greenhouse gas emissions from everything taking place on farms to getting milk to a processing plant before it goes off to grocery stores and to consumers. The majority of emissions, or the single largest source of emissions, is enteric methane from dairy cows, right? So again, this is why there's such a key focus on it, because we know it's the biggest lever we can pull if we want to lower the carbon footprint of milk. And this is also true, and even to a larger extent, for beef cattle. So this is a similar type analysis in the US of a partial life cycle assessment of US beef in terms of all the activities that go into beef cattle production before animals go to harvest. And again, the vast majority of emissions are actually enteric methane emissions that are coming from the cattle themselves. One of these wrinkles is the way cattle production takes place. Dairy production in the US is mostly animals today fed in what we call confinement. So fed in barns, fed basically diets that are mixed and balanced to meet their needs. You can kind of think about it like a cattle casserole, basically. It's a total mixed ration that we feed animals. Beef cattle is a little different in terms of its blending of grass-based production and confinement production, where we feed animals these diets delivered to them on a daily basis. So to kind of set this up, what we're looking at here is just a representation of really what the beef cattle life cycle is in the US. So animals start off on what we call cow-calf operations. So this is where the mother cows are. These animals make up the majority of the total beef cattle herd. So, you know, if you're driving around the countryside, basically anywhere in the US, you often see cattle on the side of the road, right? And those are typically cow-calf operations. The average herd size in the US is still relatively small. Roughly 50 animals per operation is the average. So those calves start off there, and when they're there, they're consuming mostly forage, right? So the calves themselves and the mama cows are basically consuming grass or hay, right, from a standpoint of what they're eating. So a lot of that cellulose material, a lot of human inedible material. Animals will then often be weaned and go to stocker and or backgrounding operations. So just two different terms there, depending on how the animals are housed. This is a kind of a flux phase of the animal's life. It really depends upon market prices, how much grass is available in the region, etc. But animals will spend more time growing on forage based diets there. And then finally, animals will go to feedlots. So typically in the US, you know, I think people assume when we have feedlots, the animals spend their whole life there, right? But this is actually just the last part of their life. So the last usually 4 to 6 months of an animal's life will be spent in a feedlot. So if we think about it from that logistic standpoint, there's methane emissions occurring in all three of these phases. And this finishing phase in feedlots is where we tend to feed more grain-based diets. So again, the vast majority of what cattle are doing is consuming forages out on extensive landscapes. And I mention all this for two reasons. One is it makes it difficult to measure methane emissions when they're in these extensive environments. And when we think about the proportion of methane, we'll show on the next slide, this is from that life cycle assessment. If we just focus on the top bar, where is the methane emissions occurring by these different stages? The vast majority of it is taking place in the cow-calf and stocker phases of the production life cycle. So it's difficult to measure in those phases, and because those animals are eating a higher forage diet, as I'll expand upon a little bit later, more forage in the animal's diet means more methane that gets produced. So the feedlot tends to make a lot less methane because there's fewer animals in that phase, but then also because of that dietary effect on enteric methane emissions. So this is, again, our challenge, and the opportunity in the cattle space is that the vast majority of methane emissions are happening from grazing animals, if we look at the whole picture of beef and dairy combined, and that's also true globally as well. So that's really where our bigger chunk of emissions is coming from a methane standpoint. So kind of transitioning into what we're doing at Colorado State University. So I'm a part of a group that is a multidisciplinary research collaborative called AgNext. So it cuts across several departments and the College of Ag, and then also the College of Vet Med and Biomedical Sciences here at Colorado State University. And a big part of what we focus on is climate smart innovation, and particularly focusing on enteric methane emissions measurement and mitigation. So this image on the screen now is just kind of giving an overview of where we do a lot of this work. So we have these climate smart research pens that we can house up to 300 individual animals at one time and get methane emissions and feed consumption data on those animals on an individual animal basis. And that'll be more clear about why that's so important in the next few slides, where I'll share some data. We also have some grazing facilities at that location, and some of these what are called green feed machines that I'll expand upon too, that we measure emissions from the animals with that can be used on grazing environments. So to sum it up, we have the capability to measure grazing cattle and animals fed in confinement at our research facility, with a particular focus on beef cattle for us. So these green feed machines, for those folks that aren't familiar, there's several ways you can measure methane emissions from ruminant animals. We are primarily using these green feed systems just because of our research objectives and the setup that we have at Colorado State University. So these are machines that are made by a company called CLOC, commercially available. And basically this principle is it is an indirect calorimetry system, similar to a head box system, if folks are familiar with those for cattle, but it's just open on one side. So as this kind of cartoon image shows, animals come up to the feeder, and there's feed in this feed bin above where the animal's head is. They voluntarily use this system, right? When they come up, there's an RFID tag reader, so a radio frequency identification tag that we put in the animal's left ear. It identifies which animal is there, and when their head is close enough to the machine, it will drop feed for them. The intent of all that is to keep the animal there for roughly 3 to 5 minutes, and while they're there, we take a spot sample of their emissions. So there's, for our particular setup, we have methane, CO2, O2, and hydrogen gas analyzers on our units. And then using those principles of, we have a known mass airflow rate, we know the background concentration of those gases and the gases that are coming from the animal themselves by difference, right? We can determine our emissions rate for each one of those spot sample visits when the animal comes to the machine. So instead of just having kind of a cartoon version, we do have a video that hopefully will work. It doesn't necessarily need the audio. But what this video is kind of demonstrating is this is a grad student put her cell phone in one of these machines last year when we were doing some research. And so this animal that's walking up is kind of demonstrating, again, they come up to this feed bin. They're wearing this little button in their left ear. That thing above the ear tag is the RFID tag. The animal puts his head in, and when its head is close enough and its tag is scanned, basically there's a little chime noise that will take place, and then green feed pellets drop. So these are just alfalfa pellets. They're just kind of basically a treat, and we kind of joke these are like the snack machines for the animals, right? So they will come up, and then the dish that the animal's eating out of and all those holes suck air continuously, collect any emissions the animal is emitting while in the machine. And that data is then used, right? That raw data is used to calculate again those emission rates. So again, that's the inside view of what these machines look like. Going here. So what does the data look like out of these machines? The nice thing about them is we can access this data in real time to check any raw data, and then on a daily basis get emission rates values back. So this is just to demonstrate one 20-minute period from one of these machines. Basically this, on the y-axis we have methane concentration. So the animal will come up to the machine, and as soon as the animal comes up to the machine, we see this usually this clear pattern of a spike in methane emissions. And ultimately what this is showing, that I had briefly mentioned earlier, cattle will have these eructation events, right, where they'll have that ruminal contraction that will expel the gas that accumulates on the headspace of the rumen out of their mouth. And for us, typically when we see this, it's like clockwork, where it is roughly once per minute we will see one of those eructation events. And then, as I've kind of pulled out and highlighted on the slide, their emissions don't necessarily go down to zero, though. In between those eructation events, right, there's still methane that's basically being respired from the animal's lungs as well, as a background basis. So again, we'll look at the area under the curve. Knowing the mass flow rate, we'll figure out emissions rate from those machines. So I'll share with you some preliminary data. The first is an experiment that was done by our master student, Swenson. This is the first data out of this unit that's really only just been around for about a year and 5 months now that we've been collecting data out of this facility. So the picture on the right kind of gives you an idea of what this setup looks like. These green bins that the animals are consuming feed out of are basically feed bins that are continuously weighed, right? So we have load cells where we can understand animal feed intake and feeding behavior on a continuous basis. Every time they stick their head in, it identifies the animal and how much feed disappearance we can attribute to each individual animal. And then those silver machines next to those green bins are these green feed machines. So each pen has a green feed machine and these smart feeds set up so we can get, again, individual feed intake and methane emissions from these animals. So this particular data set was kind of a cross-section of animals who are basically doing a feed efficiency test for these animals, which is a typical thing that's done in the industry, but then just adding on methane emissions. So to give you an idea what this data looks like, this is just the raw data. To set this up, on the y-axis we have methane emissions expressed as grams per head per day. On the x-axis, what I've done is I've taken this 2-month time frame and I've just put all the data on a 24-hour basis, so from midnight to midnight. And this is expressed for bulls, heifers in the middle, and then steers on the right. So the key thing here is there's about 19,000 individual visits over that time frame across those 6 pens for those animals. And I've identified here on the slide just kind of roughly when feeding time takes place. So the key thing here is, unsurprisingly, because animals are basically a fermentation vat, when we feed them, they eat a big meal, they put feed into the rumen, that fermentation process kicks up, methanogenesis noticeably increases, relatively quickly right after a feeding event. And so we see this diurnal pattern for animals fed in confinement when they're fed once per day. So a big part of this for us is we set our machines up so the animals can visit multiple times per day. We've done some of that work of how many spot samples do we need to get a good estimate of the animal's methane emissions. But again, this is mainly just to highlight from a measurement standpoint. Part of the challenge is when animals are fed in confinement, we don't want to just measure, for example, in the mid part of the day, right? We may be misrepresenting the total emission rate for that animal over a full diurnal basis, right? For grazing cattle, I don't have any of those slides in here, but grazing cattle, this pattern is much more flat. So those animals tend to eat many meals over time. So this is, again, this varies depending upon how we feed and manage livestock. The other thing, and this is not surprising, right, is animals, as they eat more, they produce more methane. So what we're looking at here is data for this experiment where we have methane emissions as grams per head per day on the y-axis, and then total dry matter intake, which is what DMI stands for, on the x-axis. So the total amount of feed dry matter the animals consume per day. So again, as they consume more, they emit more methane. As I've kind of highlighted here, and I'll share in a few slides, what's interesting is we find this relationship is less strong to really non-existent when we feed higher grain diets that would typically be fed to those finishing cattle, as I mentioned earlier. So one of the key things that we're interested in is understanding this variation from animal to animal. As you can kind of tell from the prior couple of slides, there's a lot of variation in methane emissions. So we're interested in understanding, can we use genetic selection potentially in the future to identify animals that are low emitters and basically select for less methane? And a key first part of that consideration is thinking about, well, what is methane also correlated to from a phenotypic standpoint in terms of how the animals express, right, these different traits? So what we're looking at in this table is phenotypic correlations, so Pearson correlation coefficients for these different traits. And these are things that are definitely important for commercial cattle producers, like how much feed does the animal consume in kilograms per day? How much do they gain? So the animal's body weight gain per day, their body weight itself in terms of how large they are, and the feed to gain ratio, right? This is an expression of efficiency. So as I've kind of summarized down on the bottom here, right? Basically, this is also not too surprising. Animals that are larger, they eat more, they gain faster, and animals that tend to be more feed efficient produce more methane per day. So this highlights this challenge, right? We want to identify animals that potentially emit less methane, but we don't want to do so independent of these other production traits, because we could definitely cost producers, right, from a standpoint of putting negative selection pressure on all the economically relevant traits for them, which would be those other animal performance traits. So there's several ways we can go about trying to correct for those different production attributes, right? So what we're looking at here is one way to try to account for this, which is basically saying regress methane emissions against mid-body weight and dry matter intake, and to try to find animals that are lower than expected emitters or higher than expected emitters based on their cohort of animals. So that x-axis on this chart is a residual mean methane emissions accounting for those different traits. So to interpret this, those dots on the scatter plot that are lower are animals that are emitting less than expected given their body weight and their feed intake. The animals that are above zero are emitting more than expected based on their body size and feed intake. And I plotted this against average daily gain on the y-axis, right? So we can kind of think about this from a standpoint of clearly animals that maybe would be desirable are those that gain, right, efficiently, but also emit less than expected, right, given their body size and their feed intake. So we can look at a subset of those animals, right, just draw a line, say, okay, those animals that are above 1.4 kilograms of body weight gain per day. And here's just kind of a summary of the highest and lowest emitters in that subset of animals, right? So they have similar body weights, they have similar average daily gain, of course, but we can see that those animals in the top and bottom emitter categories have a 33% different methane emissions in this particular subset of animals fed a high forage backgrounding diet. So again, that's just genetic selection opportunity is what we see here, is because these animals are all from the same ranch, right? And there's that much variation from animal to animal when they're all fed the exact same diet and have those similar performance traits. Even more dramatically, we've looked at the same kind of approach with animals fed a finishing diet. So this would again be that higher grain diet. And just to compare here, we had another study where we had roughly about 180 animals, and we looked at the tails of our distribution from a residual methane perspective. So we're comparing those animals' mean methane emissions at the top, methane emissions per unit of dry matter intake, their feed intake, and then their mid-body weight, right? So unsurprisingly, those last two traits are not significantly different because, of course, we were purposely correcting for those factors, so they should not be different. But you'll note, again, whether it's methane emissions per unit of feed consumed or methane emissions per head, right? There's a dramatic difference in these two groups of animals, all fed the exact same diet, all Angus type animals, actually from a common origin source, right? So we don't have all those results summarized, but we also collected rumen samples from these animals. So to look at what are those microorganisms that are actually present in these divergent groups. The key summary is definitely they have very different microbial ecologies, right? And so that's an exciting area of research, is to figure out how do we use those insights about why these animals are low or high emitters. And we can imagine a whole host of potential opportunities that can come from that, of trying to target certain microorganisms, or even an interesting area is, can you, when these animals are young, basically inoculate them with the quote unquote right type of microbes, right? And try to establish those more efficient microbiomes in their rumen at a young age. We've also done some work of just following the same animals through those different life production stages. And this is just a subset of animals, just to kind of give you an idea of what I mentioned earlier, where we know as we go through the life process and we go into a feedlot, we actually lower methane emissions, right? So this is some work that we're repeating again this year, where we measured animals right after weaning when they were in Nebraska. So these are fairly small calves eating a high forage diet. They were making 212 grams of methane per day. We grazed in the summer out on range at a USDA experimental range, and had roughly the same methane emissions at that stage. Yet when we brought them into finishing and feeding a high grain diet, and those animals, again, were much larger, gaining at a faster rate, we cut their methane emissions, right, by 60 grams per day, right? So again, one of those key things that we know we can do is by changing the diet of those animals, we can change methane emissions, but we don't want to necessarily feed all cattle high grain diet all the time, right? There's huge advantages of using forages in livestock diets, but again, this is just more of an assessment. What we're also looking at with this research is basically the repeatability of some of these rankings from a phenotype standpoint. So if an animal's a low emitter on grass, are they still a low emitter in the feedlot? At least preliminarily, unfortunately, it seems like the animals kind of re-rank themselves when they go onto different diets, which kind of points to the fact that we'll probably need to assess some of these traits on multiple diets, depending on which stage of life we want to affect. And part of that re-ranking is an interesting finding that we've seen repeated a few times, which is that with these higher energy density finishing diets, meaning again these more grain-based diets, more readily fermentable carbohydrates, the relationship between feed intake and methane emissions is quite poor, right, or non-existent. So these two scatter plots here, just from some research that we've recently completed, both are methane emissions on the y-axis and then feed intake, dry matter intake on the x-axis. So one is there's less of a range of feed intake, which is an important thing to note than like the prior chart that I shared with you all. But the reality is there just seems to be more spread, less of a relationship there between feed intake and methane emissions. And The reason this is important for us and why we're very interested in this is that even those EPA emissions estimates that I shared with you earlier, they're all based on prediction equations that really rely heavily on feed intake to predict methane emissions. So we can imagine a situation where we know animal to animal variation is not very well explained by feed intake, perhaps other factors. This could potentially create issues with the emission inventories that we have today. And approaches where folks are actually looking at trying to predict methane emissions using feed intake and then use that in genetic selection. Based on this information, right, we can see where that might cause some issues because what we observe in reality versus predicted methane may be quite divergent, again, based on these observations. So at a high level, there's many different ways that we can try to mitigate enteric methane emissions, and I'll try to keep this pretty short and then hopefully more discussion here. We can bucket these a few different ways. There's a nice review paper that came out in 2022 that kind of put them in a few different buckets. One is animal and feed management type mitigation. So one is feed processing, doing different things that we can make that feed more readily digestible and hopefully lower methane emissions per unit of feed intake, but also bigger picture systems things such as improving animal health or improving the overall productivity of the system. That's important because, as I referenced earlier, right, in the United States, for example, we've lowered the number of cattle since the 70s by over 40 million head, while actually increasing the amount of beef and milk that we produce in the United States. So clearly that has lowered methane emissions per unit of food that we as people are consuming, right? So that's a really powerful tool, and one, especially globally, just improving the productivity of animal agriculture has a huge opportunity of lowering emissions. We can also do things more targeted in terms of diet formulation. So there's evidence about, for example, tannins having an effect on reducing methane emissions, or fats, for example, supplemental fats. And then forage to concentrate ratio, as I mentioned earlier, definitely matters in terms of more grain in the diet relative to forage will lower methane emissions. And then finally, there's an exciting area, right, of more targeted rumen manipulation. Different feed additives that take different approaches to try to actually affect methanogenesis either directly or indirectly within the rumen. And at a high level, right, the two that are probably that garner the most headlines and that have so far accumulated the most research would be one, a more synthetic type approach, which is the company DSM developed a compound called 3-nitroxypropanol, or the commercial name for it is Bovaer. This is a feed additive that targets the last enzyme in the methanogenesis process. Or seaweed, right? Particularly red seaweed that contains bromoform also has a similar type effect in terms of blocking methanogenesis. And there's been some research at UC Davis in the US, and then a lot of work done in Australia on reducing methane emissions with these approaches. So the key there, though, is, as I've highlighted at the top, in the US, none of these are really commercially available yet in the US. Hopefully that will change, and part of the challenge is just the uncertainty about the regulatory process, because this is a totally new target area, right? From a standpoint of none of this work has been done in the past to be approved by the Food and Drug Administration, where it would have to be approved in the US. This particular product, Bovaer, has been approved in Europe. It's actually just recently been approved in Canada. It's available in other countries, but not yet in the US. So to sum up some of the research on these two particular products, and just to kind of highlight the reason why people are very interested in them, these are a couple quick results from meta-analyses for the two. So the first year is for Bovaer, for 3-nitroxypropanol. A summary from last year, meta-analysis looking at the mean relative difference for methane production on the left, methane yields, and methane intensity, again comparing basically no 3-nitroxypropanol versus regardless of dose, the inclusion of Bovaer basically in the animal's diet. So again, we can see pretty consistently, right, reductions in all three of those dependent variables. But the key thing for this product, it is dependent upon the dose that we feed it. So if we feed more, we get a higher percent reduction of methane emissions, right, in all three of these categories. Though there is a point of diminishing returns as we feed more of this product. And the effect is diet type dependent. In particular, as we have more forage or more fiber, particularly in the diet, we tend to get a less effective response. So for this product, there's research of higher percent reductions in, say, finishing beef cattle fed a high grain diet as compared to beef cattle fed a high forage diet, for example. So again, promising, but that's we can think back to what I shared with you earlier, that conundrum there of the dose response, or excuse me, the diet dependent response. We can think about as maybe a little unfortunate, right? Because we know more of the methane is actually coming from the forage fed animal, but it's less effective with those animals. And then this was a meta-analysis just completed recently as well from work looking at red and brown seaweed. Red seaweed is typically, as far as I'm familiar, the one that's had more research actually done on it. This is Asparagopsis species. And again, bromoform is expected to be one of the key constituents here that's really having this effect on lowering methane emissions. So you can see again a reduction in methane yield, and methane yield here is referring to methane emissions per unit of intake, right, per unit of feed intake. You can see the variation, but some of these, particularly this one Stefani study, had issues with basically the bromoform volatilizing, right? So they weren't providing the same active dose in all cases. So that's part of the explanation here. This variation in response is how the actual product is handled and if we actually have the same effective dose being delivered to animals. So I'll just kind of finish up here of we can think about all these different technical solutions, and hopefully we have more opportunities to define more technical solutions for cattle producers around the world to adopt. But a big part of the challenge is economic and other social challenges to getting these things into cattle, right? So one is we can create great feed additives, but as I mentioned earlier, because most cattle are managed extensively, that creates some challenges. That's why we're very interested in the genetics component, because we see that as a way that potentially in the long run we can lower methane emissions without having to have cattle producers deliver a feed additive out to their extensively managed cattle on a regular basis, right? We can think about the management hurdles that would potentially create. And the other key thing is the economics of it, right? Whether it increases the handling costs of how they have to manage feed, or simply just the cost of feed additives relative to premiums they may receive. So just as a quick kind of napkin math example here to try to bring that to life, I'll go through this example. So this is saying if we're a cattle feeder, so somebody who runs a feedlot, and we can look at this scenario here. What if we had a feed additive that costs us 5 cents per animal per day? So, right, sounds pretty cheap. And that feed additive will either reduce our methane emissions by 5%, so that's the dark green line, by 20%, it's the light green line, or by 50%, that's the orange line. Basically, this is saying what is the carbon price, so dollars per metric ton, that producer would need to receive for them to just break even or make money based on the percent reduction that's achieved. So you can see here with that relatively low percent reduction, right, the 5%, we have to have a very high carbon price before it economically makes sense. And an important thing here is this is assuming there's no negative or positive effects on animal performance, right? It's just all things being equal. With a 50% reduction, we still have to achieve a $32 carbon price just to break even. And if we think about current voluntary carbon prices, they may only be $2 per metric ton, right? So it depends upon the market. But again, these carbon prices for break evens in this kind of napkin math scenario are a lot higher than current carbon prices. involuntary markets. So I just throw that out there. This is where the rubber meets the road. If we can, again, from a scientific perspective, we can come up with great solutions for enteric methane, but we also have to think about how are they actually going to get adopted, and then ultimately lead to fewer methane emissions from the cattle industry in the U.S. and abroad. So with that, just to kind of sum up what our research really focuses on, one is just better understanding those baselines, right, and collecting more of this data to evaluate some of our models, testing what animals consume. We're doing some of that work on different diets and different feed additives, for example, understanding the microorganisms piece of it. So we're collaborating with those folks. As I mentioned, we don't have a lot of those results yet, but in the future, we're really excited about those opportunities of identifying which microbial populations are associated with different levels of methane production. And then finally, doing work on that genetic selection for the host or the animal itself in the long run to hopefully build methane emissions into current selection indexes that beef and dairy producers already use for methane, or excuse me, for other traits in their cattle, but build methane as one of those traits that they consider. Overall, we have this real focus on being practical, being scalable, and then really trying to think about the economic implications of any of these solutions that we're testing out for cattle producers. So with that, thanks for listening. Kind of fly through those slides real quick here. Hopefully there's some good questions, and thanks for the invite, and thanks for your attention.